Land plants originated from aquatic green algae around 470 million years ago. These first plants were small and resembled their algal ancestors and were likely constrained to moist areas close to water. These organisms had no way of obtaining water from the soil and had a limited capacity to restrict water loss from their cells. However, the challenges faced during the transition onto land transformed plants and their habitats. These challenges include gravity, exposure to higher amounts of ultraviolet light, wider ranges of temperature and temperature swings, as well as desiccation. As we cover the different aspects of plant form and function, you will notice that many of them can be understood in light of how they answer these challenges, specifically the challenge of keeping hydrated. Today, the descendants of those first land plants dominate terrestrial habitats. In particular, vascular plants. They have evolved the ability to carry out photosynthesis, grow to extreme heights, and control the gain and loss of water. If we take a look for a moment at a phylogenetic tree, we can see that land plants are part of the Bryophytes clade within the eukaryotic supergroup Archaeoplastids. This clade is made up of green algae and land plants. Land plants form a monophyletic group descended from green algae. A monophyletic group is a grouping that includes all of the descendants of a common ancestor. In this case, all land plants share a single common ancestor. Conversely, green algae are considered paraphyletic because they share a common ancestor with land plants, but land plants are not part of the green algae grouping. Land plants can be broken into two general types, the bryophytes and the vascular plants. Bryophytes include mosses, indicated in the image to the right, liverworts, and hornworts. The specific evolutionary relationship between bryophytes is not certain, but they do share several characteristics and traits. For example, they do not actively control their hydration. These plants do not form roots, though the majority of their above-ground surfaces are permeable to water. As such, they are dependent on surface water and diffusion to keep their photosynthetic cells hydrated. As their environment dries, so do they. Vascular plants, on the other hand, use specialized cells to bulk transport water and do not rely on diffusion. Additionally, they absorb water from the soil via their roots rather than from all over their surface like bryophytes, which helps to limit water loss. These features have contributed to the success of vascular plants, and they make up more than 95% of all land plant species found today. Vascular plants can be broken down into 4 main subgroups. These subgroups are largely differentiated based on how they reproduce. We have the lycophytes, which include club mosses and quillworts. Then the ferns and horsetails. The third subgroup is the gymnosperms. This includes pine trees and other conifers. And the last subgroup is the angiosperms, which encompass all the flowering plants and include oak trees, grasses, and sunflowers. Despite the variety present in vascular plants, they do share general features of their anatomy. There are 4 types of organs present. Above ground, we have the reproductive organs, leaves, and stems. Collectively, these organs make up the shoot. Then below ground, we have the 4th organ, the roots. Each of these organs are specialized for specific functions, which we'll cover in more detail in this and the upcoming lectures. There are 3 main types of tissue that make up the organs. There are the epidermis, the vascular tissue, and the ground tissue. The epidermis forms the outer surface of the plant. The vascular tissue includes the xylem and phloem and is responsible for the transport of water and carbohydrates. And lastly, the ground tissue makes up the rest of the plant not included in the other 2 tissues. This is the majority of the plant body, and the ground tissue is responsible for the majority of the metabolic functions of the plant. We'll first focus on leaves. The leaf is the principal site of photosynthesis, and its structure allows for vascular plants to perform this process without drying out. On this slide is a cross-section through a generic leaf and displays the leaf's 3 major tissues. The sheets of cells that line the upper and lower surfaces form the epidermis. Between the two layers of the epidermis is the ground tissue called the mesophyll. This is made up of loosely packed photosynthetic cells. Lastly, we have the veins, which are part of the vascular tissue that connects the leaf to the rest of the plant. Water is the resource that most often limits a plant's ability to grow and function. The majority of the water required by a plant is not due to direct usage of the water, but rather due to water loss that occurs as a consequence of CO2 uptake. The leaf's air spaces within the mesophyll are connected to the air surrounding the leaf by pores called stomata in the epidermis. As photosynthesis draws down the concentration of carbon dioxide within the leaf's air spaces relative to the concentration of carbon dioxide in the outside air, the difference in concentration between the inside and the outside of the leaf causes the CO2 to diffuse into the leaf, replenishing the supply of CO2 for photosynthesis. The cost to a photosynthesizing plant is water vapor loss. If CO2 is able to diffuse into the leaf, water vapor can diffuse out. The rate at which water vapor can diffuse out is much greater than the rate at which CO2 can diffuse inward. Water is lighter than CO2, and it diffuses 1.6 times faster. Additionally, on a sunny day, the difference in concentration of water vapor within the leaf relative to the outside of the leaf is much greater than the difference in carbon dioxide concentration. This means that several hundred water molecules can be lost from the leaf for every molecule of CO2 acquired. The ultimate result is that the water cost for photosynthesis in the air is large. For example, an acre of corn can lose 4,000 gallons of water a day. That is equal to about 95 bathtubs of water. This evaporative loss of water vapor from the leaves is called transpiration. To combat the loss of water due to transpiration, the leaf continues to pull water in from the vascular system, which absorbs water from the soil. On top of this are structural adaptations that further limit loss of water. One such adaptation is the cuticle. The waxy cuticle is secreted by the epidermal cells on their outer surface and can be seen on the previous slide. The cuticle limits water loss from an epidermal cell, but it also prevents carbon dioxide diffusion into the cell. As mentioned earlier, small pores called stomata in the epidermis allow CO2 to diffuse into the leaf. The stomata serve as an adaptive compromise between the challenge of water loss and obtaining the materials for photosynthesis. Stomata can be numerous on the surface of a leaf, up to 1,000 per square millimeter. However, they are very small, and less than 1 to 2% of the leaf's surface is actually covered by these pores. Stomata are able to help maintain the balance between water loss and CO2 uptake by opening and closing in response to factors that affect these two competing pressures. Stomata function as hydromechanical valves that can open and close. Each stoma consists of 2 guard cells surrounding a central pore. The guard cells can regulate the size of the pore by shrinking or swelling. The 2 guard cells are joined to each other at their ends, and they have cellulose fibers that wrap around each cell. This means that when the guard cells swell, they expand in length rather than width, causing them to bow apart and thus opening the stoma. As the guard cells shrink, they then close the stomatal pore. Guard cells control their volume by altering the concentration of solutes in their cytoplasm. An increase in solute concentration causes water to flow into the cell by osmosis, whereas a decrease causes water to flow out of the cell. The accumulation of solutes inside guard cells requires an expenditure of energy in the form of ATP, which drives the uptake of solutes across the plasma membrane. In contrast, closing does not require energy expenditure. The default state of a guard cell is resting or closed, and this is the state that conserves the hydration of the leaf. Stomata respond to light and signaling molecules so that they are open when it is favorable for photosynthesis to occur and closed when the leaf needs to conserve water. The next organ we'll focus on is the stem. Here on this slide is a cross-section through a sunflower stem. A layer of epidermal cells lies along the surface of the stem. This layer of cells encloses thin-walled parenchyma cells in the interior. Parenchyma cells are part of the ground tissue. Vascular tissues are found within the ground tissue and lie in a ring near the outside of the stem. These vascular tissues are part of a continuous pathway that extends throughout the plant's 4 organs. The outer vascular tissue, called the phloem, transports carbohydrates from the leaves to the rest of the plant body. The inner tissue, called xylem, transports water from the roots to the leaves. We'll look into these 2 types of vascular tissue in more detail in the next lecture. If you have any questions, please let me know via the Q&A discussion board.